okay. That's okay. Let's just go ahead and address any questions you may have, and even if they're not about lab, I don't mind. So I'm gonna go ahead and put a whiteboard app in case I need it. So okay, you can go ahead and ask anything you want. Is the test still going to be due on Saturday? Probably yes. I have no problem. Yeah, because this is the deal. Why? It's because we finished chapter 14. Started chapter 15, we should be able to wrap it up on Thursday as far as going over the lecture material. So I have no problem with it. Also because we have, let's see, this is lecture test 5, and then we really have only 4 weeks left. And one of those weeks is Thanksgiving week, which is next week. So after this week, we have 3 weeks left to cover chapter 16, 19, and gram-positive and gram-negative pathogens. So I don't want to push this fifth test due date any further. I think we need to wrap it up so that we can concentrate on on the last part. And also because lab, there's a lot of things due on lab also. So the last weeks of the semester are very busy. And that's why I don't want to change due dates. I've actually changed the due date of something, which I'm going to tell you right now, one of the lab project things. But so as far as due dates are concerned, for lecture, test 5, which is chapters 13 that we have already covered, 14, which we have already covered, 15, which we started, so we need to finish on Thursday, and a little bit of 18, which is PowerPoints only. I think there's about 25 PowerPoints at the most. We should be able to finish these no problem by Thursday. So due date is Saturday, which is the 21st, I believe. Take a look at that. Okay. Yeah, the 21st. Okay. That will leave us with about 3 weeks to cover the lecture test 6 material, which is going to be chapters 16, 19, and then gram-positive and gram-negative pathogens. And that one will be due on 12/10. So that's lecture due dates. That leaves us with the last 3 weeks to cover the material for test 6. And when it comes to lab, actually, well, for the lab material that's left, this is what we're going to have. Oh yeah, we have a practical 2, which is due on 12/10. So, and that is why originally I had one of the unknown assignments due this Saturday also. I have changed, however. So we have, first of all, Lab 11, which is optional. And we're going to talk about Lab 11 next time. I think I said we're going to talk about it today. I'm going to leave it for next time because Lab 11 is due on 11/28. And this is an optional assignment. Then we have an assignment called the unknowns. And this consists of 2 virtual labs. Each is worth 50% of the total unknowns. Okay, one is the virtual lab is called, yes, 20 points each, and total of 40 points for unknowns. That is correct. So The first one is the virtual lab is called unknown identification. It's already, and I should probably go ahead and let me go ahead and put it up on eCampus. But give me a second. I want to do just a second. Let me check something out real quick. Perfect. Okay, so I'll go ahead and open up the I'll open up eCampus so that we can see where all this is located. So this is unknown identification, which we're going to go over today. It's up on both. Yes, it should be up on both. I put it up on both over the weekend. So unknown ID, it's due. Now, it was originally due on Saturday. I have changed the due date to the 28th. And there was I'll explain the reason why I wanted it done a little bit quicker, but that's okay. We'll go ahead and push it back. And then the other one, which is medical unknowns or medical microbiology, I think the actual title is, that one is due on the 5th, December 5th. Okay. The reason why I wanted at least one of them done as quickly as possible is because the material that we cover in both these virtual labs is going to be part of the practical exam. So I wanted it covered as quickly as possible so that we can, so that you can start getting ready for the practical. But that's okay. There's something else in this exercise. There is an optional. So let me go ahead and put optionals in red. This is optional. There is an extra credit clinical case, which is worth 10 points, I think. I have to look. Oh, we'll look whenever I'm there. The extra points are going to go towards the unknowns. I'm pretty sure it's 10 points. Again, I could be wrong, but I'm almost certain. And it's going to be added to these 40 points right here. All right, we have that also. Again, this is optional. That is it for the lab, and then we have the lab practical, obviously. And then lab practical too. Now, that is due 12/10. I will open it by the 5th, so we'll have hopefully I'll open it before the 5th, so we'll have at least a week to get it done. So a couple of days before, I'll open it on the 3rd. Never mind. This is no, we end on the 12th, on the 10th. Sorry, we end on the 12th. Then I'll open it 7 days before, so I can open it on the 3rd. Have a week to wrap it up. I'm going to start posting the study guides for Lab Practical 2 as I continue to. So I'm going to be posting the study guide corresponding to Lab Practical to Lab Reports 7, 8, and 8 this week, and then I post for 9 also this week. 7, 8, and 9 will be posted this week. Lab Report 10 was due today, so as soon as I grade it, I'll post the study guide for that one. And then I will need to post the study guide for these 2 exercises right here also. So it will be posted as separate document, each study guide for each lab report. So let me show you where all this is on eCampus. Let me go ahead and share application screen. And I guess all right. Let me see, make sure you're there now. Yep. Oh, no, not yet. Oh, there we go. Perfect. Okay. So if you go to laboratory, and I am on the 71020 section, but it should be on the other one also. You scroll down. This is lab report 11, which is the optional one. Here we have unknown virtual lab assignments right here. When you click here, we have biochemical test PowerPoint, which we're going to go over today. identification of unknown bacteria report. The PowerPoint we're going to go over today, the biochemical tests, will be helpful for the identification of unknown bacteria for these exercise. Here's the virtual lab. Again, everything is due on November 28th. So note the new due date, November 28th, as opposed to November 21st. So we have extended the due date for identification of unknown bacterial report. Then we have medical microbiology identification, which is similar to identification of unknown bacteria, except that it deals with gram-positives. Identification of unknown bacteria looks at gram-negatives. Medical microbiology identification looks at gram-positives. And some of the same biochemical tests apply. And then here is the extra optional clinical case, worth 10 extra points towards the unknown exercises. And this one is due on 12/5 also. I don't have a due date here. I need to put that up there so that all of the unknown exercises will be due on the 5th of December. Okay, give me a second. I missed your question. Let me go back there. 10 each or 10 total? Give me just a second. Okay, I'm not sure about 10 each or 10 total. What do you mean by that? You said 10 points for the unknown. Oh, no, 10 total. I see what you're saying. No, 10 total. So the unknowns are worth 40 points total. 20 points come from one of the virtual labs, so identification of unknown bacteria. 20 points come from medical microbiology. So extra optional clinical case will go to the total, not to each of these two. Yeah, so it's 10 points total, which is still significant because it's a fourth of what the total grade is anyway. So it for the most part ends up being 10 extra points period, which added to the whole bucket anyway. So that's actually a pretty good extra points. Okay. Yeah, hopefully that's what you meant. I think that was your question there. Perfect. Okay. So let's see what else. That's how we're gonna wrap up the lab. Again, I'm gonna be putting the study guides, and what I'm gonna do with the study guides is I'm gonna put the document at the very top. I will probably bring all these labs that we've already done to the bottom so that we have it organized with what's left at the top. Okay. And that way you don't have to scroll down so much to find all that material. So it will be similar to what we did here, how we have it all in chunks. It will be the same way. It will be A, B, C, D, and F, and A will be lab 7, and the next one 8, 9, 10. It will be like that. Okay. So, any other questions? And if more questions come up between now and Thursday, we can address lab material or lab issues on Thursday also, so we don't have to just talk about it today. Okay, so this is what I have as far as PowerPoints. I have the biochemical tests. So let me go ahead and address those. And if you have questions as we're going through these, like always, please don't hesitate to just ask them. So in a face-to-face lab, what happens is that we towards the end of the semester, about 4 or 5 weeks before the semester ends, we have an exercise in which students get a bacteria and they have to conduct a series of biochemical tests to identify the genus and the species of the bacteria. So this is as close to that, give you a little bit of a taste of the kinds of tests that need to be done in order to identify an unknown organism. So one of the first tests, or the first procedures that need to be done to identify bacteria, is to determine the gram reaction of the organism. That's crucial. That's the number one thing that we need to do. So gram reaction and the shape. So gram reaction, actually, let me go ahead and go back to a whiteboard. I think that's going to be helpful. Gram reaction and shape is one of the first things that get done. There's a reason why I want a whiteboard. Okay, there we go. Okay, so to ID microorganisms. Gram reaction. So a gram stain will tell you whether you have a gram positive or a gram negative. So let's say that what you have is a gram negative. Okay, so you do the gram reaction, you decide you have a gram negative, so you're going in this direction, your organism is not gram positive. The next step is to determine the shape. So shape, you could have cocci, you could have a rod, and yes, there are other shapes like spirals, but in most medical microbiology, these are the two shapes you're looking at. Most medical microorganisms or microorganisms that are medically significant are either going to be cocci or rods. So let's say that, again, through the gram stain, you determine you have a gram negative rod. Okay, so you're going again in this direction. So this is no, this is yes. So this is where you're at. The next tests that are done with gram negatives are typically carbohydrate fermentation tests that allow you to determine what kind of sugars can the microorganism ferment. And first of all, you are assuming the organism can ferment. So facultative anaerobes, which are a lot of our medically important gram-negative rods, can ferment sugars. Strict aerobes, on the other hand, which some of the medically significant gram-negative rods are strict aerobes, they cannot ferment sugars. So sugar fermentations are very important to know what groups you're looking at. And that will be probably another one of those tests that will be done fairly early on in the identification process. So the sugars that are typically tested are going to be glucose, which is the main sugar that most microorganisms are going to consume and be able to catabolize, followed by lactose, mannitol, and sucrose. Those are the most important sugars. So let's say that you want to see whether the organism can ferment glucose. So ferments glucose. And you say no, yes. And you've determined that your organism does ferment glucose. So this is where you're at right here. From there, you want to know whether the organism can produce hydrogen sulfide. Okay, H2S producer. You're going to say yes or no, and let's say yours is no, and you move on in this manner until you eliminate all the possible organisms and you get to your organism. So for example, when it comes to whether you have a fermenter of glucose or not, you will be, if it's no, then you're eliminating organisms like Pseudomonas, Alkaligenes. And over here, you get a whole bunch that could ferment glucose. Being not an H2S fermenter, you're eliminating the ones that, I'm not fermenter, I'm sorry, producer, you are eliminating the ones that can produce H2S. So let's say Proteus and Salmonella, and I can think of any other one that makes H2S off the top of my head, but those will be eliminated, and you are here with E. coli and Enterobacter and a list of the possibilities. And you continue to do tests that will differentiate those organisms that still remain on your list and eliminate some of them via testing until you end up with only one. So that's the kind of procedure that you're going to be doing in the virtual lab. And you will have a dichotomous key very similar to the one I made here for gram-negatives. And as you follow this dichotomous key, you will be able to, in each of the cases presented, identify your organism. So pay attention to the dichotomous key that they are giving you in the virtual exercise. And if you follow it, there's no way you can get anything wrong. Well, I guess the only way you can get something wrong is if you don't read the test correctly. But we're going to go over this test now, so you should not read the test incorrectly. Yes, okay. The problem with the lab manual is that it is assuming that we're doing this exercise in a lab. And you're right, there is a table at the back of the lab manual that gives you some of the results for a lot of these organisms, and that would be helpful too. Absolutely, yes. You're absolutely right. Yeah, I forgot about that table. Yeah, especially if you pay attention to which ones are gram-negatives while you're doing this exercise. And for now, put aside the gram-positives because this first exercise is gram-negatives only. Okay. So yes, if you do that, the lab manual can also be very helpful. So that's essentially what the virtual lab takes you through. So it is important to understand what are the biochemical tests that we're going to be doing and how to read them. It is also important to understand them because this is going to be part of the practical. All of these tests, the principles of the test, the how to read them, what's a positive, what's a negative, all this will be part of the practical. Some of the kind of media that is very useful to identify bacteria, especially in a clinical setting, in a medical lab, is what we call selective and differential media. And this is the principle behind selective and differential media. And again, this is very, very useful in a clinical setting, because in a clinical setting, what you're doing is you're getting a specimen from a patient. Oftentimes, the specimen is loaded with microorganisms. The specimen could be something like a stool sample. It could be a respiratory discharge, sputum, or it could be a wound discharge. So all of these examples I'm giving are examples of specimens that are going to have lots of microorganisms in them. And you have to be able, the medical microbiologist has to be able to distinguish or separate normal flora from the organism that is causing the infection, knowing that the microorganism that's causing the infection could also be a normal flora that has overgrown. But nonetheless, you have to separate a specific microorganism of interest from everybody else that happens to be there. So, and because in a medical setting you are rushing against time, you want to be able to identify that organism as quickly as possible. It is very useful, and keep in mind, growing a bacteria takes at least 12 hours, at least, if not 18 or 24. So we're talking a whole day or 2 days before you actually have growth. Forget about even identifying. So it is very useful to be able to do both at the same time. grow the organism, separate it, and identify it. If you can do all three things within 18 hours, within 12 to 18 hours, you're ahead of the game when you have a patient waiting for the results. And that's where selective and differential media come in. These are specialized media that were typically designed to be used in medical labs that allows you to do just that. Separate organisms, grow them, differentiate them all at once. Okay, and you'll see what I mean when I start giving you examples of these media. All right. Okay, so a selective media is a medium that allows you to grow one group of organisms over another group. So the media is going to allow one group to grow, but not the other group. For example, it will be a media that allows the growth of gram-negatives, but not of gram-positives. Or vice versa, allows the growth of gram-positives, but not of gram-negatives. That's selective. And then if the media is differential, that means that the group that grows, we can separate those organisms that grow and put them into groups. For example, gram-negatives and can grow in the medium, and then you can differentiate or you can separate those that can ferment lactose from those that cannot ferment lactose, okay, within the same medium by just looking at how the organism grows. So that's the differential part of the medium. Okay, so examples of selective and differential media: mannitol salt agar plate. We're going to look at that one. MacConkey's EMB, which is not on the list, but we're probably going to look at that one too. Okay. All right. So first one we're going to look at is MSA plates, which stand for mannitol salt agar plates. This is a selective and differential media, which will be useful for the next virtual lab, not this one, because it selects for gram-positives. It selects for specifically the genus of gram-positive cocci, Staphylococcus. And then it's going to differentiate among the Staphylococcus. So the media was designed to allow Staphylococcus to grow and kill or not allow the growth of gram-negatives and other organisms that were gram-positives, but they were not Staphylococcus. This is the way it works. The selective part of the medium is based on the plates having salt in them. So that's the name mannitol, which is a sugar, salt agar plates. So mannitol is a sugar. Salt is what allows the differential part of the medium. Salt, the medium has 10 to 15% concentration of salt. Contrast that to what other media has. Other media is going to have 1% at the most. Salt is typically 0.9 to 0.85 salt concentration. So this is 10 times more than what other regular media has. Remember from chapter 4, I believe it was, and also from, I believe it was lab report 6, that Staphylococcus are a genus of bacteria that can tolerate high salt concentrations. They are halotolerant. You can also call them osmotolerant. What that means is that they tolerate a high concentration of salt. They prefer not to have it, but if they do, it's not going to kill them. They will just grow. Maybe may take a little bit longer for them to grow, but then they're still going to grow. Okay, so Staphylococcus genus are halotolerant. So the high salt will not stop their growth. That is the basis for selection. So if we receive a wound specimen, And some of the most common organisms that cause wound infections are Staphylococcus. But there will be other organisms in that wound that are not going to be halotolerant. What that means is whatever organism grows on the medium is a Staphylococcus. Anything in the specimen that was not a Staphylococcus is not going to grow. So, for example, Micrococcus luteus could also be in that wound, but is not a staph, so it will not grow in the MSA plate. A gram-negative E. coli will not grow on this because the salt concentration will stop the growth of anything except Staphylococcus. So that's the selective part of the medium. The differential part of the medium. MSA plates are going to differentiate between Staphylococcus that can ferment mannitol and Staphylococcus that cannot ferment mannitol. If you have the lab manual, go to the last page and look at the Staphylococcus. There's 3 of those, and look at which of them can ferment mannitol, which ones cannot ferment mannitol. And this is how the medium works. The medium has the sugar mannitol incorporated. It also has a pH indicator called phenol red. Phenol red is a dye. Most dyes are pH indicators. They change colors depending on the pH. In the case of phenol red, at pH 7, phenol red is red. As the pH becomes more acidic, less than 7, the color of the dye changes to yellow. As it becomes more basic, with a pH higher than 7, the color changes to pink, bright pink. Okay, now, if a bacteria ferments a sugar, when bacteria ferment, they make acids. That's from chapter 6. So, if the bacteria can ferment the sugar mannitol, a waste product of fermentation that the bacteria will begin to secrete out into the medium is going to be acids. Those acids are going to change the pH of the medium towards the acidic part, turning the medium yellow. One of the Staphylococcus that can ferment mannitol is the Staphylococcus aureus. So if you have a Staphylococcus aureus growing on an MSA plate, the plate will turn yellow. The fact that the organism is growing tells you it's a Staphylococcus. The medium turned yellow, so that tells you it's a Staphylococcus that can ferment mannitol. It could be aureus because aureus is one of the staph that can ferment mannitol. Here you have a bacteria growing on the medium, so you know the bacteria growing is a Staphylococcus. However, the medium has not turned yellow, which means whatever Staphylococcus is growing in this medium cannot ferment mannitol. Okay, well, if you look at what Staphylococcus cannot ferment mannitol, one of them is epidermidis. So that tells us that the organism growing in that plate may be Staphylococcus epidermidis. If nothing grows on the plate, that means that there was no staph in the specimen that was used. So that's how MSA plates work. You should know the principle of the MSA plates. Why do they work? What are we looking for? What's in the medium? How to read it. So that at practical, there will be examples of MSA plate results, and you should be able to interpret those results. Questions about the MSA plate? Another selective and differential media is MacConkey's Agar. And this one is for gram negatives. The medium has dyes that are toxic to gram positives, but not to gram negatives, so that gram positives do not grow in the medium, gram negatives grow in the medium. So that is the basis for the selective part of the medium. So it selects for gram negatives. The differential part, those gram negatives that grow in the medium can be differentiated in the basis of whether they can ferment lactose or not ferment lactose. And that's based on the color of the colony. This is what the medium looks like originally. It has a couple of dyes in there. One dye is eosin, which is a red dye. Another one is methylene blue, which is a blue dye. So that those dyes deter the growth of gram positives. So bacteria that grow in the medium are going to be gram negative. So just by growing them, you know these two organisms are gram negative. Okay, so right off the bat, you know the gram reaction of the organism. Whatever was put in this plate was probably a gram positive, probably because it didn't grow. Okay, now out of the gram negatives that grow, you can differentiate between those that can ferment lactose and those that cannot ferment lactose. If the organism can ferment lactose, it will put out acids, and the acidity is going to allow the bacterial cell to take up the dyes that are present in the medium. The dyes are red and blue. When you mix red and blue, you end up with a purple color. So that's why you end up with purple colonies from the organisms that can ferment lactose. If the organism is not fermenting lactose, there are no acids being produced, and the cells in those colonies are not taking up the dye, so they do not turn purple. So purple colonies tells you the organism is lactose fermenter, so the A is a gram negative that can ferment lactose. We call lactose positive. B, organism B, is a gram negative that is lactose negative, cannot ferment lactose. So you can take a look at the back of the lab manual chart, look at all the gram negatives there, and realize that some can ferment lactose, others are lactose negative. If you look at the chart, organisms like E. coli can ferment lactose, for example. We call them those coliforms. The organisms that are typically found in feces are coliforms and are lactose fermenters. So you should know the principle behind the MacConkey plate, how it helps you differentiate between the lactose positives and the lactose negative, gram negative, and it's selective for gram negatives. Another medium that is important in this case to differentiate, this is not a selective medium, this is a differential medium only. So this is the BAP, which stands for blood agar plate. Differential is not selective. Okay. Blood agar plates are agar plates that have 2% sheep red blood cells added to the medium. Sheep red blood cells, or blood cells, period, are highly nutritious to bacteria. So this is an nutrient-rich medium, which encourages the growth of pretty much any organism. So this is not a selective medium. Anything can grow on blood on this agar. It's not going to select for anything. All bacteria can grow on blood agar because blood is a nutrient-rich medium. However, what we're looking for in this medium is the ability of the bacteria to destroy the red blood cells, something that we call hemolysis. There are 3 types of hemolysis. Beta hemolytic bacteria are the bacteria that produces enzymes that completely destroy red blood cells. We call them beta hemolytics, and they're going to completely burst the red blood cells in the medium to where the red blood cells will be gone, completely gone. Alpha hemolytic bacteria secrete enzymes that partially destroys red blood cells. So there will still be blood cells in the medium. There will only be partial destruction of these cells. Gamma hemolytic bacteria cannot destroy red blood cells. And this is a term that we're trying to get away from, calling something gamma hemolytic, when in reality, these are not hemolytics. But the virtual lab is calling them gamma hemolytics, so we're going to go ahead and continue calling them that way, even though we're trying to stay away from keep calling them that way because they're not hemolytics, but that's okay. So they do not destroy red blood cells. These plates is showing you all 3 types of hemolysis. These right here are the beta hemolytics. And let me explain what you're seeing. The bacteria growing in here is putting out, yes, group B strep is beta hemolytic. Group A strep is also beta hemolytic, but yes, absolutely. So, and streptococcus is, hemolysis is very useful in identifying streptococcus, as you will find in the second virtual lab, medical microbiology for gram-positives. So, no, it doesn't. It does not stand for beta hemolytics. This was a group. These groups were created back in the 50s, I believe it was, 1950s. They're called the Lansfield group to identify streptococcus. And this was a microbiologist called Rebecca Lansfield. And she did work on the streptococcus, and she classified the streptococcus, and she put them into groups. And they're called the Lansfield group of classification of streptococcus. Group A, Group B, Group C, Group D. There are some that are not part of the any group, but no, it doesn't stand for beta hemolytics. Yeah, a little confusing, but that's just the way it is. But many of the streptococcus are hemolytic. Like Group A streptococcus are hemolytic. So when beta hemolytics are the ones that completely hemolyze red blood cells, and that's why the medium has turned clear in this area, is because the red blood cells are gone. As a matter of fact, the medium is completely clear. If you were to put your hand on the other side of the plate, you will see your hand there because all the red blood cells are gone. Alpha hemolytics or partial hemolysis, which is really it's not really hemolysis. What is happening is that the enzymes are oxidizing hemoglobin. So it's really messing with the hemoglobin more than with the red blood cells themselves. Nonetheless, we call them hemolytics. Alpha hemolytics, you see this greenish tint to the area. I would call it a greenish halo. So if you look at one little colony right here, like this one right here, you'll see a green color around the growth. So that greenish color is alpha hemolysis. A lot of the organisms that live in our throat are alpha hemolytic. So it is not unusual to see alpha hemolytics coming from a throat specimen. what we're looking for in a throated specimen are the beta hemolytics. Those are the potential pathogenic ones. And then gamma hemolytics, which are non-hemolytics, will not harm or change the blood at all. So in the second virtual lab, you will do hemolysis tests on some of these gram-positives to be able to identify them. So this test is especially important for gram-positives, hemolysis tests. Okay, now biochemical tests are useful for both gram-positives and gram-negatives and allows us to identify bacteria. I will give you the names of the test and the name of the medium used for the test. So this is the citrate test. The name of the medium, Simmons citrate. What we're looking for, the purpose. And you should know purpose, indicators, how to read a positive and a negative reaction for the lab practical tool. The purpose of the test is to test for the ability of the organism to use citrate as a source of energy. If the organism can live on citrate only. And citrate is a carbon compound. The organism must be able to break it down. Yes, like the citric acid cycle. Yes, it's part of the citric acid cycle. It's part of the Krebs cycle. So if the organism can take that citrate and continue it on and break it down to make energy, then it will be able to live in the Simmons citrate medium. Otherwise, it will not be able to live in there. So the medium has citrate as the only source of food. It has an indicator called bronchithimal blue. This is a pH indicator, which is going to be green initially under neutral pH. Now, when the organism breaks down citrate, the end products are going to be basic compounds. So, in contrast with the fermentation tests, which gave us acidic products, the citrate test gives us basic pH, high pH products. Okay, the medium with the bronchithimal blue will change colors from green to blue if the organism is making basic products. So that's how you tell a positive reaction. So if the organism can utilize citrate, it will produce basic products that will change the pH of the medium from a neutral to basic, and the indicator will change from green to blue. So this is a positive reaction. The organism is making basic products, so the bronchithimal blue changes colors to blue. This is the initial, because that's a negative. This is the initial color of the test tube, and nothing changes really if the organism is negative. Okay, so you should know that a blue is positive, a green is negative. Urease test is another test that is very useful for identifying microorganisms. In this case, we want to know whether the organism has the ability to produce enzymes called ureases that break down urea to ammonia. Again, same principle, it's just pH indicators. In this case, the pH indicator is the same one we use for the MSA test, phenol red. Remember I said phenol red is red under pH 7. So 7 pH, the medium is red. We said that it turned, if the medium was less than 7, it turned yellow. If the medium turned more than 7, basic, it turned bright pink. If the bacteria makes ureases that can break down ammonia, ureases that can break down urea, not ammonia, urea. So urease, urea turns to ammonia when it's broken down. And ammonia, you probably know, is basic. So the medium, the uninoculated medium, we start out with a reddish orangey pH 7 medium. Inoculate the organism. If the medium turns bright pink, the organism is breaking down the urea medium, and the pH is increasing because of the accumulation of ammonia, giving us the bright pink. That's a positive reaction. If instead the media turns yellowish or orangey, it's negative. It's a very straightforward test. You don't need to know the pH indicator in the medium. You need to know what we're looking for and what's a positive negative reaction. And I did mention one of the first tests done to identify organisms is sugar fermentation. There are many ways to test for sugar fermentation. One of the most common ones, though, is to use a test tube that has the sugars, medium that has the sugars, and it has phenol red added to it. Again, pH indicator, phenol red. So these are called phenol red sugar tests. So we have phenol red glucose test, phenol red lactose test, phenol red mannitol test, phenol red sucrose test. You can add any sugar that we want, but those four are the most common sugars that are tested. We're looking for the ability of the organism to ferment the sugar. So first of all, the organism must be a fermenter. If it cannot ferment, nothing is going to happen. So assuming the organism can ferment the sugar, if it ferments, it's going to get acidic products as an impact of fermentation, which means the phenol red is going to change from red to yellow. So a positive reaction is yellow. That indicates acidic conditions brought about by the fermentation of the sugar. A negative reaction, anything that is not yellow. That means orange, that means red, that means pink. Anything that is not yellow is negative. So here you have not yellow, that is negative. Yellow, positive. Yellow, positive. This right here is a little bit of a what you have here is an organism that has made so many acids that the phenol red has been completely reduced. And you have a faint yellowish color. You have gases being produced. Actually, this one is producing gases also right there. You can see gases in that collected in that little tube. So this is a tremendous fermenter that produces so many acids that, again, that phenol red has been completely reduced. Most of the time, you just definitely can see that yellow color. So there you have a positive or a negative phenol red sucrose. Regardless of the sugar, the way it's red is always the same. Yellow is positive, no yellow, no orange, red is negative. Okay, halfway there. Okay, this is medium created for a medical microbiology lab. This is the principle. Again, you're racing against time. You want to be able to grow and identify the organism as quickly as possible. So, what if you have media that allows you to do more than one test at a time? That's what the SIM is. The SIM is a medium that allows you to test for hydrogen sulfide production. That's what the S stands for. Indole, and I'll tell you what all this is in just a second, and motility. So with one test tube, you can test for 3 things. So within 18 hours, you'll be able to acquire enough information to be able to identify the organism. Okay. H2S production. Essentially, what we're looking for here is the ability of the organism to produce hydrogen sulfide. An organism that produces hydrogen sulfide is one that can break down certain amino acids that have sulfur in them. There's 2 amino acids that have sulfur in them. Cysteine is one of them. And if the organism can break down these amino acids to make energy from them, then the sulfur in the amino acid will be released as hydrogen sulfide. Very few organisms can do this, though. So that's a kind of a test that few organisms will give you a positive for this test. Nonetheless, the medium has an indicator for hydrogen sulfide production. And let me go ahead and erase what I wrote here. Okay. H2S, when it binds to iron, it creates a black precipitate. So the medium is inoculated with the microorganism. It is incubated for 24 hours, 24 hours. So you need at least 24 hours after putting the organism in the medium to be able to read the test. So it's not immediate. You have to grow the organism and let it do what it needs to do. Let it produce, break down amino acids, ferment sugars, whatever they need to do. That takes time. That takes about 24 hours, if not 48 hours. So after the incubation period, you take the tube out of the incubator and you look at it. If there is a black precipitate, that means the organism was making hydrogen sulfide. The hydrogen sulfide bound to the iron already present in the medium, and that's where the black precipitate was made. So the iron is the indicator that indicates, detects the presence of the hydrogen sulfide. So there's a positive for hydrogen sulfide production, there's a negative for hydrogen sulfide production. That takes care of the S in the SIM media. The SIM media also tests for indole. That's where the I comes in. Indole looks at the ability of the organism to break down an amino acid called tryptophan. There's 20 naturally occurring amino acids. Tryptophan is one of the 20. If the organism can break it down to make energy out of the tryptophan, then it will be, what remains, the waste product will be pyruvic acid. Pyruvic acid and indole. Those will be the two end products of tryptophan breakdown. Again, you inoculate the medium, incubate 24-48 hours. After incubation, you read for hydrogen sulfide production. Okay, no black precipitate, so this is H2S negative. Then you read for indole. You read for indole by adding drops of a reagent called COVAX, which is going to detect the presence of indole. You put the COVAX in. COVAX has a yellowish color unless indole is present. If indole is present, COVAX changes to red. So you have here H2S negative, indole positive. H2S negative, indole negative. Okay, so that's how the SIM media is read for hydrogen sulfide production and indole. The M stands for motility, and that one is a little bit trickier. Essentially, all it is the medium, when it's inoculated, it is inoculated with a needle. So we introduce the organism by stabbing the gel with a needle, and the needle carries the microorganism. Okay. And then you look as to whether the microorganism grew away from that stab, indicating it swam, it was able to move, or whether, well, in this case, let me go ahead and clean this up. In this case, you don't see the stab anymore because the microorganism swam away from it and it grew all over the medium. Here, however, you can see that the growth is limited to the stab where you put it. So the organism is non-motile. Here you have a motile organism. So this might not be as useful to you guys in this virtual lab, but it's part of the SIM medium. So these are some of the results that we can get with the SIM medium. Here you have an organism that H2S negative. You cannot really barely see a stab here, which means the organism is growing all over. So it is motile. And you see the red on top after adding COVAX, telling you that it's indole positive. Here you can see the stab. So it's non-motile, H2S negative, indole negative. Here you have an indole negative up here on top. H2S positive, you cannot see any stabs. However, it turns out all H2S positives are motile. So just because it's H2S positive and all H2S positives happen to be motile, you can say that it is also motile. Here you have an indole negative, H2S negative, and motile. This one is motile, H2S positive, indole positive. So again, when you're reading the SIM test, you have to read all three tests. Okay, moving on. Nitrate test or nitratase test, typically just called the nitrate test. This is what we're doing. The principle of the test. We want to know whether the organism can reduce nitrate. Nitrate is NO3. If the organism can reduce nitrate, which means that it can use nitrate in the electron transport chain as a final electron acceptor instead of oxygen, it's going to reduce it to nitrite. Some organisms can reduce nitrite further to ammonia, and some can even go further and reduce it all the way to nitrogen gas. So if the organism can reduce nitrate, there are three possibilities as far as positive reactions. The organism can reduce the nitrate all the way to nitrite, to ammonia, or nitrogen gas. If the organism cannot reduce nitrate, then NO3 will still be present in the medium after incubation. So this is how the test is done. You take the broth, inoculate it with the organism, let it grow for 24-48 hours, and after growing the organism, you're going to test for the presence of nitrite. We test for the presence of nitrite by adding reagents A and B to the medium. So, is NO2 present? Add reagents A and B. If the medium turns red, adding reagents A and B, then that means that yes, NO2 is present. And the organism is nitrate positive. It can reduce nitrate to nitrite. If the medium does not turn red, you cannot say that this is a negative reaction because it could be that the organism has reduced nitrate to ammonia or it has reduced nitrate to gas. So you can still not say it's negative. It could still be positive. So now you do the next test. You add zinc. Zinc detects the presence of NO3. Zinc binds to nitrate and turns it red. So if you zinc and the medium turns red, that means NO3 was never reduced. Nitrate is still there, present. It has not been changed. This is a negative result. If there is no red after zinc, that is a positive result because that tells you NO3 is gone. It may have been turned to ammonia, it may have been turned to gas, it doesn't matter, it's gone. So the organism was able to reduce the nitrate to either ammonia or gas. So the organism is positive for nitrogen. So essentially, that's how the test is done. So no red after A and B reagents, and then you turn zinc, then you get zinc and it turns red. That is a negative result. So negative after zinc, red after zinc is negative. This one turned red after adding reagents A and B. This one is positive. These two are positive. This one, no red after adding reagents. We added zinc, and the media does not turn red. That means it's positive. So this is positive. These are positive. This is negative. Okay, so that's the principle of the nitrate test. You should know the principle, and you should know what is a positive reaction, what is a negative reaction. Bioescalene test will be helpful in the next lab in the gram positives. Essentially, can the organism break down bioescalene? If it can, in the medium, after incubation, it will turn a black precipitate. If it cannot, it will remain the original color. So it's a straightforward test. MRVP is another test important for gram negatives. This is actually two tests in one medium. The medium is called the MRVP broth. And with the MRVP broth, you can test for MR and you can test for VP. MR stands for mixed acids, or actually stands for methyl red. So MR stands for methyl red, which is the indicator in the medium that we're going to add. But we're testing for the ability of the organism to ferment glucose and make mixed acids as end products of fermentation. So you inoculate the medium, incubate for 48 hours. After incubation, to see whether the organism has made acids from glucose fermentation, we add the pH indicator methyl red. If the medium turns red, that indicates acidity, and that means positive MR. Low pH, positive MR. If the medium doesn't change into red, is negative for MR, which means the organism is not making a mixture of acids after fermenting glucose. Okay, so what is it doing after fermenting glucose if it's not making acids? There's the other possibility. The organism could be making butanediol as the end product of fermentation. We test for butanediol with the VP test. So, inoculate the medium, incubate. After incubation, add reagents A and B, Barrett's reagents. If the medium turns red after about 30 minutes after adding the reagents, then it is positive for butanediol. So we say the organism is a butanediol fermenter. It makes butanediol. Yes, this was in chapter 6, actually. We explained this test in chapter 6. Yep, this is negative. So essentially, this is what we saw in chapter 6, right here. These are tests that allows us to determine products of fermentation, of glucose fermentation. So an organism that is MR positive and BP negative means that it is a mixed acid fermenter. It produces mixed acids as waste of fermentation. E. coli is an example of an MR positive organism. Some organisms produce butyridiol among the products of fermentation, the end products, the waste products of fermentation. Those will be BP positive. Enterobacter is an example of an organism that makes butyridiol. It will be negative with the MR test, positive for the BP test. So those two tests are run together, and the organism will either be positive for one and negative for the other, or vice versa. So it's useful to separate among these fermenters. Never run both at the same time. Oh, I see what you're saying. Both. Okay. No. Actually, some organisms can produce both. A few. It's rare, but there are a few organisms that will actually give you a positive for both tests. Most of the time, however, it's either one or the other. But yeah, there are a few genera that are able to make both. So if you look at the chart, look at the proteus. Among the proteus, some of the proteus, there are two proteus in the chart, Mirabilis and vulgaris. And I believe one of them can make both, I think. My memory serves me. Catalase test is another one of those tests that we talked about in chapter 4, actually. The ability of the organism to have catalases. So organisms that had catalases were the strict aerobes. Actually, it's probably easier to see the ones that don't have catalases. The strict aerobes, facultatives, actually that's macroaerophiles. These organisms were catalase positive. Organisms that were catalase negatives were the aerotolerants and strict anaerobes. These are catalase negative. Catalase was one of the enzymes that protect us from oxygen radicals. So any organism that can live in the presence of oxygen has catalases. Organisms that do not like oxygen or are not well protected from them are catalase negatives. The way we test for catalase, incredibly simple. You drop hydrogen peroxide on the organism. Catalases turn hydrogen peroxide into water and oxygen. So the bubbles we see are oxygen bubbles. So if you drop hydrogen peroxide and bubbles appear, the organism is catalase positive. If you drop hydrogen peroxide and no bubbles appear, the organism is catalase negative. So it's a very easy test. Okay, almost finished here. A couple of more tests, I think, and that's all. Yeah. Okay, KIA test. All right. This is similar to the SIM medium in that this is more than one test in one tube. It's called Kliger's iron agar test, and it actually testing for glucose, lactose, and H2S production. It tests for 3 things at a time. So it's used for the detection of carbohydrate fermentation in bacteria. Glucose, lactose are the 2 carbohydrates tested. And then on top of that, it tests for hydrogen sulfide production. This is the way the sugars work. The indicator in the medium is phenol red. That's the pH indicator in the medium. We're already familiar with this pH indicator. Notice that it says that lactose is present in a concentration 10 times that of glucose. So we have 1 part glucose to 10 parts lactose. So most organisms are going to use glucose. The organism lives on glucose, and glucose runs out. Ferments the glucose, is making acidic products. The glucose runs out. After the glucose runs out, very quickly, if the organism can't ferment lactose, it will continue to live fermenting lactose. If it cannot ferment lactose, then that's it. It has no more food. Okay, so if the organism can ferment the glucose only, the medium will turn yellow at the bottom because there is such little glucose that only enough acidity will be made to turn the bottom the organism began fermenting yellow. But it was not able to ferment the lactose. So this will be glucose positive, lactose negative. Here, the organism was not able to do either. So glucose and lactose negative. Let me give you more results. Okay, take a look at this one. This one, the organism turned the whole medium yellow. That means it was able to ferment the glucose positive, and then it turned to the lactose and began fermenting it, lactose positive. Here you have black precipitate telling you H2S production. The medium has iron just like the SIM. So this is H2S positive. You can see the top is yellow, so it's lactose positive. And oh gosh, a test for the lac operon. Yes, it can be used for that, for the ability of the organism to turn on the lac operon and make beta-galactosidase and utilize lactose. Yeah, exactly. Actually, yes, specifically, because only that bacteria that has a lac operon will be able to utilize the lactose. To utilize the lactose, I'm trying to think here, to ferment the lactose. In order to ferment the lactose has to be cut into, lactose is, I know we're going a little bit off of the mark, but that's okay. Lactose is made by, is a disaccharide, is made by glucose and galactose. So beta-galactosidase splits lactose into glucose and galactose. Glucose goes directly into glycolysis, and the organism makes energy from that glucose. Galactose, the lac operon, has another enzyme that turns the galactose into glucose, and it goes into glycolysis. So for the organism to be able to use the lactose, it has to have the lac operon. So absolutely, lactose positives have lac operons. And yes, this is taking that principle because the initial glucose allows the organism to live, to grow, and then when the glucose is gone, then it's going to turn on the lac operon and begin utilizing the lactose. So this guy definitely has a lac operon. This one right here is an H2S producer. All H2S producers are glucose positive. So even though we cannot see the bottom, whether it's yellow or red, it doesn't matter. All H2S producers can ferment glucose. This one, on the other hand, is different because it doesn't have the yellow top, it has a red top. So this tells you the organism is lactose negative. It is H2S positive, which means that it's glucose positive also. And this is the control. So that's how we read the Klicker's iron agar. You read the slant, you read the bot, and you read for H2S production. And H2S production can be detected with the KIA agar or SIM agar. Okay, finally the oxidase test, which you will do on this lab. essentially, it wants to know whether the organism has a cytochrome C oxidase in the electron transport chain. So this is essentially one of the electron carriers of the electron transport chain. And some bacteria has cytochrome C, others don't. So specifically, Aeromonas, Pseudomonas, Neisseria are oxidase positive, whereas members of the Enterobacteriaceae family are oxidase negative. So we want to separate between oxidase positive and oxidase negative organisms. Oxidase positives are belong to this genera of bacteria, either Aeromonas, Pseudomonas, or Neisseria. Everybody else is Enterobacteriaceae family. The test is very easy. You just put a little bit of the organism on a filter paper. A reagent is it. So you put the filter paper, you put the bacteria on the filter paper, and here you can see the bacteria was put right here. Add the reagents. If if there is oxidase, if there's cytochrome oxidase present, the organism, the filter will turn purple. If there is nothing present, there will be no purple. It's a pretty easy test. Okay, so this should be able to take you through this first virtual lab. The gram-positive virtual lab uses a lot of the same tests, plus a few more that we're going to see next Monday. But the ones that we've seen today should take you through this virtual lab without any problem. Any questions? Yeah, let me go through the actual lab report real quick. Just make sure there's nothing there. Give me a second. This is it. Yeah, okay. It says 25 points. That's because there is I guess 5 extra points or something. This is just 20 points though. All right, so what this virtual lab report gives you is, first of all, a sample exercise, a sample result. Most of these cases is going to ask you for the direction and shape of the organism, and then where the specimen came from. So in this example, the specimen is a wound sample. These are the tests done. The first test done is the oxidase test. In this case, the result was that there was no change in color after adding the oxidase reagent, which that tells you that is oxidase negative, is a negative reaction. And that tells you that these are the organisms that are not eliminated. So one of these organisms is the final identification organism. The next test, second test done, was a lactose fermentation test using MacConkey's Agar. It showed growth of clear white colonies. The interpretation of that is that it's negative for lactose fermentation. These are the organisms that are negative for lactose fermentation. So now we eliminated E. coli, we eliminated Heterobacter frundi, Enterobacter aerogenes, Klebsiella pneumonia. Vulgaris was not eliminated, Diffemerium was not eliminated, Marcescens was not eliminated, Salmonella soni was not eliminated. So this is still a possibility. Next test is H2S production. It was used a KIA agar, which had a black precipitate, giving you positive for H2S production. That eliminates Marcescens and soni, leaving you with Vulgaris and Diffemerium, Salmonella Diffemerium. Fourth test is a urea test. There was a change in color from clear to pink, giving you a positive reaction for urea, leaving you with Proteus vulgaris. So that is the organism that was identified from the wound sample. And that's essentially how you're going to do it. The next will be a water sample. The organisms will be different for students, so not everybody's going to get the same results. Keep that in mind. You need to identify the organisms that you're given. So the program, the virtual lab, has several possibilities as results for each one of these cases. So case 2, case 3, case 4, and then case 5. So we have 5 cases. Okay. So keep that in mind. Keep in mind that not everybody's going to get the same results. So I will go by the tests, the results of the tests. You need to explain the results of the test. So for example, let me go back to the second case. It asks, through your test, which enzymes, if any, were found in this organism. Okay, so if the organism is urea positive, then the organism has the urease enzyme. If the organism is catalase positive, then it has the catalase enzyme. If it is oxidase positive, it has the cytochrome oxidase enzyme. If it is nitrate positive, it has the nitratase enzyme. If it is indole positive, it has the tryptophanase enzyme. You should be able to come up with at least one enzyme for each organism, at least. You may not have, you may not do the urease test on your organism, so you would not know whether it has the urease or not. You may not do the nitrite test or the indole test, but pretty much every time you're going to do the oxidase test and the catalase test. So at least one of these, you should be able to find out whether your organism has that enzyme or not. Is the organism a fermenter? If so, which sugars are being fermented? Okay. And explain the medium use for each of these tests. What medium was used? What is the principle of the tests? Any reagents or indicators used in test 2? Okay, never mind, test 2. So the second set of tests. Same thing for the third set of tests. Give the medium use, the reagent use. And if there is a fourth set of tests, then answer the same for each test. So for the second, third, and fourth set of tests, you need to describe the medium used for the test, what was the principle of the test, any reagents that were used on the test, etc. And it just keeps repeating the same thing over and over again. You do the same with 5 different cases. And that's essentially the extent of the exercise. Okay, any questions?